Hello everyone, this is Emanuele Martini. I'm a biology image analyst since 15 years, and I'm a researcher in cancer research in an institute in Italy, but I pass most of my life in Uganda in Africa, and I'm part of the African Bioimaging Consortium. Today we will just have a brief introduction to the microscopy image analysis, in particular in biology. So the objective of today, it will be to understand how a digital image is acquired, which are the image file formats, the software used in digital images, in particular the one for biology, the important understanding of what is the image intensity histogram, basic step of digital image processing, noise and background, and signal to noise ratio, and the signal intensity, how the signal intensity can be increased, and what is a find object through the segmentation and threshold. And then we discuss which parameters then you can quantify and analyze from your biology experiment. First thing to understand is how to define digital images and what they are and how they are produced. A digital image is the result of two kinds of digitizations. The first one is the sampling, and it is related to the coordinates and to the space. So how many pixels, how many space you have to define your analog image, the real object. And then you divide your image in squares called pixel. Each pixel then is quantized with an intensity. This is the second digitization that is called quantization. So starting from an analog image, the real object of interest that you are acquiring with a camera, with your microscope, you sample it in coordinates, like a matrix of points called pixels, and each pixel is then quantized in terms of intensity. There are different ways to capture light. Most we can divide them in cameras and photon sensor and detectors. So you can have CCD cameras, CMOS cameras, and PMT detectors. So you have a cathode, and then you capture the electrons through the anode, and then you transform it in voltage to an analog digital to converter, and then finally you have your intensity pixel quantized, the one we saw before in the second step of digitization from an analog image. Then, of course, you have two types of camera in biology. One is monochrome, and the other one is color. So you can capture fluorescence, for example, with monochrome cameras, and you can capture tissues, slides from patients from tissues with an RGB color camera. One important thing to consider is always how you save and you export your images, and in particular, which kind of compression you use or you are not using. There are two types of compression. The first one is lossy, so it means you are losing information. The second one is lossless. So obviously, when you use the first one, the lossy compression, you realize a file size that is smaller and you get some information lost. And this is the example of JPEG and GIF. And this one is really not good for science topics and science analysis. So you should never use it for quantization in the terms of science. And there are some lossless compression, PNG and TIFF, and this one, they preserve the information when they are restored. So just to be a little bit more clear on it, JPEG is lossy. You can widely use it outside of biology and science. Then GIF, for moving images, a very small, extremely reduced size. Then there's PNG for sharing on the internet. Some data is retained. You can have transparency options, but these are lossless compression. But the best one for our scope in biology microscopy and in science microscopy and science images is the TIFF. Lossless compression, and you can fully restore resolution and the image metadata, like the resolution of your objective, the laser you used to acquire your images, and all the acquisition parameters. So this is the best way to store microscopy. There are really a lot of open source free software for image analysis and scientific image analysis. So they are free and open source. You can use them, change and share in modified or modified form. There are some of them are more used than the others, but this is the main ones. Fish in particular, Coupa, Self-Profile, and Elastic. They have all some pros and cons. We are not going to discuss it, but you can imagine some of them are more useful for handling a lot of data. Some of them are more useful for handling fluorescence and cell culture data. Other ones are more for pathologists and then for tissue and patients' life, and the other ones are more for machine learning and deep learning analysis and implementation. Now we will introduce one of the main protagonist character of our of image analysis, and this is the image intensity. The image intensity histogram is the distribution of the intensity of pixel of your image. So on the x-axis, you have all the possible intensity values that it depends on the camera and on the sensor you are using. And then on the Y, you have the number of pixels, the count of the pixel with a certain value. So at the end, it's really the distribution of the intensity of your images. So the range of the histogram depends on your detector. It goes from darkest to brightest bigness, and then the same image can be taken without the brightest pixel being saturated. So what is saturation? Saturation is when you obtain pixels that have the maximum level of your dynamic range of your detector. What is the maximum range of your detector? It's depending on the bit depth. So on the camera or the sensor or the system you're using to get and acquire your images. And the bit depth is the number that defines the number of grayscale levels you can have on your image. So for example, if you have 2 bit, the number of grayscale levels you can get is 4. Then you have 64 for 4 bit, and then 8 bit, for example, you can have 256 intensity level. And just consider the grayscale always include 0. So for example, for 8-bit images, your intensity scale will go from 0 to 255. Just a few examples about what it means have different bit depth. For example, here we are taking the eye of the person in this picture, and if you only have 2 bit, you can only have 4 levels of gray, so you have an image like that. As soon as you increase your number of bit, you have more gray levels, and so you have more resolution power in terms of. This is, for example, with 4 bit, and so you see that we can have 16 levels from 0 to 16. And then, for example, when you pass to 8-bit histogram, you have 256 gray level. That means that you can represent your eye like this, and you start to see a lot of gray shades of gray levels in terms of intensity. Just remember, it's an image histogram. It's the distribution of intensities of your image. So, in the number, the count of pixels with a certain intensity. Now, it's important to understand which are good histograms, which are good acquisition, which are bad acquisition, and which are the things you have to pay attention when you acquire your image. In this case, for example, we have an underexposed image because it doesn't make use of the full dynamic range that you have in this case of an 8-bit camera, so you can see from 0 to 254. And then you can see the levels used by your acquisition are not using all of them. For example, you are starting to have some intensity around 35, and then you stop at 190, something like that, and you have a lot brightest intensity left out, and also before around the darkest ones. So you are underexposing your image, and so you have a not full dynamic range histogram. This situation is even worse because is when you are overexposing your image, and then you obtain a lot of pixels that are higher than the maximum level emitted by your sensor. In this case, always we are always using an 8-bit camera sensor, so our maximum would be 255, and as you can see, there are a lot of pixels in this area. This is a little bit worse than the situation before, so overexposed histogram is a little bit worse than underexposed, because when you have saturated pixels, these ones on the maximum level, you cannot do anything in image analysis processing to retrieve them. They all look the same, so you cannot distinguish them anymore. Instead, with the underexposed, you lose maybe some information because you are not getting the whole possibility of your camera, but at least you are not saturating pixel, and you are not having pixels that you cannot distinguish their intensities. In an ideal world, this is a correctly exposed image and histogram. So if you can, it's always better to try during acquisition to have a correctly exposed histogram that use the full range of your sensor. Now we will talk a little bit usual image and flow. So the first thing usually you do, considering the goal of your biological experiment and the reason why you are acquiring your images, is do some preprocessing. So image preprocessing is a way and algorithms to remove noise, to subtract background, to improve the contrast. Usually, this is done to have a better detection, better so-called segmentation. Segmentation is a term, technical term we will use sometimes during this presentation. It means when you recognize the borders, the boundaries of the object of interest. In our case, can be nuclei, mitochondria, vesicles, cytoskeleton, whatever is the object you want, this biological object you want to recognize in your object. So usually, yeah, there are different methods to find the objects. The most famous one and basic one is the thresholding. Then you can refine your detections, and then you can identify your region of interest, so your object in terms of boundaries, and instance them in the sense that you can identify single object with an identity. So what is the, for example, the nucleus number one, the nucleus number two, and so on. And then after that, you can extract features and parameters to distinguish and to inspect your biological experiment and get answer to your biological research question. Preprocessing is necessary because in our field, in biological field, you can have background, ghost signal for many reasons. Even if we talk about overexposed and underexposed, sometimes so signal is necessary to keep the cells alive. So you have to have a trade-off with background or signal, out of focus area, and then what you really want is to obtain a good signal to noise ratio. That means that the signal of interest is much higher than the noise you have. So also this helps to increase the contrast. Preprocessing helps to increase the contrast, improve in some way the signal to noise ratio, and in particular to it's very important when you need to identify objects of interest in the sense as we were saying of filaments, lamellipodia, cells, nuclei, mitochondria, really depending on your experiment. So good quality should display good contrast, and then they allow you for better segmentation than the subsequent steps like a quantification, measurements, features, and so on. Three aspects are the main ones that affect in contrast, and then there are signal, background, and noise. Let's just have a few slides about what are those and try to understand a little bit more about them and how they play together to give you a good or a bad signal to noise ratio image. So first of all, it's nice to point out the difference between noise and background. So background is non-informative signal, so stray light, autofluorescence. A lot of biological samples have autofluorescence. So when you acquire them, you obtain the signal you want, and then but also non-informative signal that is going to be autofluorescence, non-specific staining. So maybe you have another staining, and then you get some fluorescence and some lights also from there, and dark noise. And this contribute to signal to background ratio. Most of the time, we can assume it as a constant input. then you can really subtract from the final image most of the time. Noise, on the other case, is randomly corruption of an image due to the moreover to the uncertainty of photon detection. So it can vary, and then is not so good to assume it constant for your input, so it can there's a variation when after, even if you give to it as input signal constant, it comes from the microscope itself, and you have different way to quantify it and see it. So there is the detector, the signal amplification, the sensor you're using, the camera you're using, and so on. For example, you can see this maybe with this image and this quantification. We take a single bead and we acquire them and quantify the single bead 21 times. So we acquire them 21 times to see how is the signal and how is the signal around this line in orange you can see here. What you can see if you measure them and you quantify them 21 times, sorry, is that you have something like that. So you have your signal in orange, and then around the signal you have some non-constant variation, and this is really the noise because it's not constant, and you can evaluate it maybe as we have done, taking multiple images of the same object, and then instead you have background here on the bottom right. That is the non-specific signal, like a level of non-specific signal that is always there, and you can, as we said, you have a signal to noise ratio. I would not enter too much into the details of the noise you can have, but they are, you can relate them to the camera, to the photon count, and to the photon and the emission light properties of lights. Instead, the background, as we say, are signal that is not informative. For example, no specific staining, autofluorescence, stray light, noise called dark noise, and you can also calculate a measurement that is the signal to background. Now, just a slide about what aspect contributes to signal intensity. So, because you remember, no, you have a signal to noise ratio and signal to background ratio. So, and you remember from the first slide, you need to have an high ratio in this case because we want a very well contrasted image that is more easy to maybe find object, to quantify, to interpret. So you want, in the first place, reduce the noise in the background. In the second place, or the same time, increase your signal. The signal, as you maybe know from the other courses in this series of introduction to microscopy, it can be from the abundance, from the brighter dyes, from optical concentration of the dye. Just when you increase those things, you have to pay attention to the so-called photo bleaching. That it means that you are exaggerating with the light and you are irreversibly destroying the fluorophore and you cannot recover it. So of course you can increase a lot the signal, but you have always to pay attention to photo bleaching, and also in another way also to the viability of your cells, of your sample, if they are live cell imaging. So you also have to pay attention to don't give too much intensity to your cell and kill them if they are alive. How can improve it? Also to improving your light path. So the configuration, like which microscope you are using, which components, the filter, the pinhole for microscope confocal, and maybe this is why it's important to ask expert or read very well the papers about which kind of configuration of the optical path they are using. Then of course you can play with the illumination, always pay attention to the photo bleaching, increasing gain of the detector in the confocal microscopy, and the exposure time of the camera. In a part of photo bleaching, when you increase intensity, gain, and exposure time, you always have to pay attention to, now you can also say, to saturation, because if you really put a lot of illumination and light, you really risk to go in the overexposed histogram that we say is not really good, in other words, it's bad for image quantification because you are going to have a lot of saturated. So just a few words about You have two kinds of approaches to enhance your image. The first one are called the point operation. So you are manipulating directly the histogram and the values regardless where they are in the image. You are just changing them depending on their intensity. So you are going to change the brightness and contrast. Or and this is a linear way to do it. So you are changing brightness and contrast. We refer to a way to change with a linear mapping, with a linear function, the intensity of your pixel, depending on the intensity of the pixel itself. Or gamma correction when you want to perform some more refined changing in the intensities, um, pay more attention if the intensity are higher or lower. You can have different kind of correction, but it's very important to understand that these ones are point operation. So you are just changing intensity values regardless of where the pixels are in the space and whatever. Just they consider just the intensity itself. On the other hand, filtering are algorithm applied to a group of pixels, to a neighborhood. So those group of pixels are called kernels, and they are different from point operation because they consider a group of pixels together, and then of course the intensities, but this group of pixels, not just the pixel itself, but the pixel with the neighborhood. So a group of pixels is changing, considering the pixel around. For example, one famous one is the Gaussian blur. It's can be useful, this kind of approaches, to have a better segmentation step, an easier segmentation. What is detection and segmentation? We already said something about it, just a few slides to be more precise and to fix our in our head and the concept. Thresholding means distinguishing between meaningful signal and signal that is not required for the analysis. So in a certain way, we can also say foreground intensities from background intensities. Here you can see a threshold applied to these images. What you can see, it's it's this threshold tool from Fiji, but is like this for in all the image analysis software. You are fixing a value, in this case 29, and then you are saying all the pixels above the intensity of 29 with an intensity above higher than 29 are of some interest, and this meaningful signal, and in your image they are shown in red. And also you are considering all the pixels below than 2055, that is the maximum for your camera. As you can remember, with an 8-bit camera the maximum is 255. So all the pixels between 29 and 255 are considered meaningful signal, so foreground. All the other ones are considered background, that they are not considered, and you can distinguish it applying a so-called threshold. And this is a binary image. Usually, this process produces for you a binary image when you have interesting pixel and not interesting pixel. So 1 for interesting pixel, 0 for pixel of not interest. And so 1, or in another way, white where you see foreground pixel, and black when you have not interesting pixel. And so these regions, how you can get them? The process to get them is called segmentation. So segmentation allows you to detect different objects, to recognize them, and usually we refer to them as a region of interest, sorry, and you can use that to then do your quantification of single independent identified objects. For example, here on the left, we can, as you can see, we put a threshold, in this case around 100, and so you are getting all the objects with, in this case, coins above, with an intensity higher of 100 as foreground and background, as we have done before in the slide, in the previous slide. And now this is where we were before. So this is the thresholding process. Now we need a way to identify single object, and this is called segmentation. So you can see now each coin has its own identity, its own label. You can even call it label image, and its own boundaries. And these ones are the so-called region of interest. So they are now independent, different objects recognized through the process of segmentation. Of course, you can apply the same for the same concept for biological images, and then you are recognizing cells, vesicles, and nuclei as independent objects, and so you are segmenting it. Obviously, you can do a lot of measurement. You can divide them in mainly two types. The morphological measurement, maybe you are interested in the cell area, in the elongation. So if the aspect ratio is I or 1, depending if it's elongated, how much they are similar to a circle, if the convex whole area, so the area taking the whole convex outside considering all the boundaries of your image is similar, bigger than the real area of your object. And this kind of morphological measurement can tell you a lot about the biology under under your experiment, under your condition. I just want to cite one example. Tumoral cells usually have an higher aspect ratio, they are more elongated, and so on. So, and then you can, of course, depending on your biological experiment, you can really have different conditions that brings to different morphological measurement. And obviously, you can have intensity measurement, like in this case, you can do cell count live versus dead if you are able to put dyes and staining that identify that in your sample. Then the cell size, the average intensity. So maybe if you have put an antibody on some protein, you can have a proxy to a protein level in different conditions, in different cell. You can compare intensity bigger versus smaller cell. For example, size of the brightest cells, size of the clear also in the cells, maybe, circularity, different morphological. And with that, we finish. It's a very introductory course. All the slides you have seen, they can be expanded in very many other slides or the other talks. For example, the background subtraction, the noise reduction, the filtering, the image pre-processing, the way to segment cells. But this is just an introductory course, and I thank you for your attention, and we are open to questions here and also in via email or contact on to ABIC